Thank you for the organizer for letting me present today. So thanks, Matt, for the introduction. So a little bit of introduction of myself. So right now I lead the laboratory, the Center for CRISPR Target Discovery at the IGI. So I worked on various pool and array screening projects over the years. And today I'll be presenting to you some of the examples of using CRISPR in repression and activation screens. But first of all, I would like to introduce a little bit more of the institute itself. The Innovative Genomic Institute was founded by Jennifer Doudna, who co-discovered CRISPR gene editing technology. And the institute itself is divided into certain areas in terms of engineering technologies and how to use that to advance human health. And also there's another part of the institute that focuses on engineering microbes to fight climate change and increase crop yield and also public engagement. And so the Institute has various initiatives or centers that are focused on different parts of CRISPR technology development. For example, we have a consortium that is advancing CRISPR delivery in vivo, and that is driven by advances in lipid nanoparticle technology or peptide-based RNP delivery. In addition to adenovirus-based delivery modalities. And there's another similar part of my center that is ATG, Advanced Translational Genetics, that is building arrayed iPSC models to study diseases. And there's a very important initiative that is primarily driven by our Director of Technology and Translation, Dr. Fyodor Urnov, who has a strong interest in advancing N-of-1 CRISPR therapies. And this is sponsored by the Danaher Corporation. This is called a Beacon for CRISPR Cures. And that leads to a really important news this year. Baby KJ at the Children's Hospital at Philadelphia, who was able to receive a CRISPR therapy within the first 6 months of his life to correct a liver enzyme deficiency. And through this work, we just have announced that the Chan Zuckerberg Initiative is sponsoring the founding of a new center for pediatric CRISPR cures. That is a joint venture between UC Berkeley, IGI, and UCSF. So my center focus on pool CRISPR screens to advance target discovery. So this is a path where we are looking for novel gene targets that are implicated in diseases such as cancer and neurodegenerative diseases. And hopefully that will translate to new therapies that can move into the clinic and address unmet medical need. So if we can advance the screening part and also lead to the characterization of those gene targets, and hopefully there could be a way to phenocopy those target repressions by either looking for existing drugs targeting those gene targets or having new compound developed towards those that currently do not have a drug for. And today's talk will be centered around two topics, using two screen examples, one using CRISPR-I screen, another using CRISPR-A screen. And we can see some of the kind of like technical hurdles that we have to address along the way, and what is the best way to utilize these novel libraries. And the first collaboration we have is with Steve Oshuler and Nani Wu at UCSF. Their research focuses on targeted treatment resistant in cancer via non-genetic mechanisms. So their expertise is in high-content image-based array drug screens. So if we think about treatment resistant in cancer, there are two different ways to think about this. First of all, the tumor themselves may have a mutation that cannot be addressed by the targeted therapy. And so that is primary resistance. However, there are also acquired resistance that emerges through treatment. And what they're interested in is this persistent evolution where the tumor responds very well initially, but there's always some cells that are left behind that leads to drug tolerance in the long term. And this is what we can see a lot with targeted therapy in lung cancer or colon cancer, where patients respond really well initially. However, after 6 months or so or longer on treatment, the tumors start to come back, and usually they are on exactly the same place where the primary tumor was. And that they either have like a selective pressure that causes them to eventually grow out, but they are still able to respond to the treatment very well initially. And it's important to understand those mechanisms to address treatment resistance in cancer. So their initial research is focused on EGFR inhibitor, alatinib, in EGFR mutant-driven non-small cell lung cancers. When they did an arrayed culture media screen, like looking at what are the amino acid and cytokines and growth factors that would have an impact on pro-persistent phenotype, they found that interferon gamma can both promote and mitigate persistent phenotype depending on the dose. And so to look into that, they then did another screen using interferon gamma in that context, in addition to alatinib, and looking at what are the existing drugs. And this is using a compound array library to look at what are the inhibitors that can be added to interferon gamma to specifically induce cell death or lack of proliferation. And so what they found is this PRMT inhibitor, that this is arginine methyltransferase enzyme that regulate epigenetic gene expression. And so once you identify that target, you can then think about combining the two together, interferon gamma and this inhibitor that target PRMT enzymes, and that can actually reduce the persistence of alatinib. And this is using a relatively high dose of alatinib over a 2 month period. And so this is a really nice example to see how target discovery can work towards better treatment. However, the lab is interested in developing genome-wide perturbation to have a more unbiased way of identify treatment-resistant target. And of course, that in the non-small cell lung cancer field, KRAS-driven lung cancer has a much higher unmet need. There's only two currently FDA-approved targeted therapy for KRAS. And hopefully there will be more. And so it is important to understand the mechanism of persistence that are based on gene expression changes instead of the genetic mutations that arises out of the treatment. So in that case, this is more suitable project for a CRISPR-I screen. Since we're thinking about like drug treatment, in terms of drug development, perhaps it's most unlikely you have a drug that can inhibit some gene target that is at 100%. So a gene repression approach is probably more suitable for this type of venture. However, the challenge for this type of screen is that sometimes they are interested in using like really harsh treatment. So what we're talking about is only 5 to 10% of the cells that are surviving the drug treatment to induce this persistent phenotype. And what we need is a much smaller CRISPR library. And the first generation of CRISPR libraries are huge. In this kind of like a summary of how we're going to screen this looks very straightforward. It's a drug versus no drug comparison. And you can do this in like 3 or 4 weeks. However, if you think about the practical consideration, if your library size from, say, like CRISPR-I library, which is the the first generation, it's over 100,000 library diversity. And if you want a coverage of 1,000-fold and account for the MOI at infection stage and your IC90, so only 10% of the cells surviving that, once you accounted for all the losses along the way, you want the number of flags that you need to start with is huge. And during the point where we are doing puromycin selection for the library incorporated cells, we estimate that we need close to 300 flags of T225. So this is not very practical. So what we're looking for are something a lot smaller. And that's around the time where we have the ultra-compact CRISPR-I library that came out of Jonathan Weissman's lab. And so that's the technology that we'll talk about more in this part of the talk. And so what's the difference between CRISPR-I and CRISPR knockout? In this scenario, we're using dCas9, catalytically dead Cas9 that do not cut the DNA. And so in a lot of the times, we'll consider it to be less toxic due to the wild-type Cas9 that normally will induce double-strand break and a DNA damage response signal. And CRISPR-I can target non-coding regulatory gene regions instead of the knockout version where it normally targets early exons. However, the guide RNA selection is different. So there need to be a different criteria for that in terms of library construction. And since the genes, this is gene repression, not gene knockout, there won't be complete gene silencing usually. So this is actually more comparable to shRNA library type of screen. However, the gene knockdown itself, some people consider them to be more representative of drug inhibitor effect. So that's why sometimes people may want to use CRISPR-I instead of CRISPR knockout in the screen. And to improve CRISPR-I signals, people have tried mainly two different pathways. So they either engineer more effective repressor domain. For example, in this scenario, the initial gene repressor domain, CRAB, is taken from ZNF10. And then they compare that to other type of transcription factors that contain CRAB domain. And they identify that actually you have this ZIM3 CRAB domain that seem to be more effective in terms of producing a more inhibitory effect. If we look at an example cell surface marker, CD81 in this case. And so they were able to produce a stronger inhibitor effect using this domain. And that's what we'll use. In the first generation of CRISPR-I library, Luke Gilbert's lab had, so when he was a postdoc at Jonathan Weissman's lab, he did library construction using essential genes and proliferation genes to look for where is the design window for the guide RNA. And so from the initial kind of 5 base pair plus and minus at the transcription start site, the TSS site, they design around 1,000 guide RNA per gene for CRISPR-A and CRISPR-I, and trying to figure out where is the sweet spot for the guide RNA to land on. And from that, using machine learning algorithm, they're able to identify for CRISPR-I a window that is slightly downstream of the transcription start site that seem to be optimal in terms of inhibiting the gene function. And for the CRISPR-A version, there is a window of around 500 base pair that is slightly upstream of the transcription start site that seem to have the most signal. However, at the first generation of genome scale CRISPR-A, CRISPR-I library, they still have 10 guide per gene. And that library is still really big, around 210,000 elements in the library for each CRISPR-A and CRISPR-I. So then Max Holdback then used computational method, machine learning to look at the guide performance and coupling that to some of the other annotations on UCSC's genome browser. For example, Phantom 5, the transcription start site, that is coupled also to promoter regions. And this give information on nucleosome accessibility. And for CRISPR-I, CRISPR-A, that is important. And so when they did a comparison of the second and first generation CRISPR library screen, they can see that the second generation in blue has a much stronger signal, especially that they also have a different region of target compared to the first generation. The first generation target, the annotated transcription start site, and the second generation target more on the non-promoter usage. So with that, they were able to rank the performance of the guide RNAs, and they can combine the first 5 into a much smaller library. That's also divided into 7 sub-libraries by their gene function. So this already were able to narrow the library size down to 100,000 elements. And in the third generation, Joseph Rapogo, again in John Weissman's lab, devised this method of coupling 2 guide RNA together, and that will give more effect to the gene repression. And so using this dual guide system, they validated the design across 6 cell lines. And so this is the design of the vector structure, where you have from one vector, you have 2 guide RNA being transcribed, and both guide RNA targeting the same gene. So you're getting double the strength of repression and activation. And in this bottom panel here, we are getting much stronger dropout when we're using the dual guide RNA library system versus the single guide. And so this is one way to then increase the strength of these dCas9 species in the bound to the DNA. And so they then divide up the guide according to their performance. Say like guide 1 and 2 can be a pair, guide 3 and 4 can be a pair, and guide 5 and 6 can be a pair. So you can have the option of using just one pair, or you can have 3 of them combined into one. If you are considering an ultra compact library, this will be 20,000 different species. If you want to use all 3, which will have 3 data points, then the library size is 60,000. So that's already a lot smaller than the initial 200,000 elements in the first generation library. And so this is kind of the structure of what the guide RNA being transcribed. So there are 2 versions, one with an index barcode between the 2 guide RNA. So each guide RNA is transcribed from its own promoter. And this is kind of like the sequencing strategy that they use that will be slightly different between whether you use the index barcode version. And so one issue that they have found is that occasionally there will be decoupling of the 2 guides, and that sometimes can be like 30% of your reads that do not have the perfectly matched guide 1 and guide 2. And so I always like to see what the sequencing amplicon look like. And so this is a snapshot of what the sequencing amplicon you will submit. And so it requires both for read 1 and the reverse read 2, custom read primers, and also a custom index primer that would read sample index that was added on the reverse primer during the amplicon generation. And so we'll go back to the guide decoupling issue a bit later after we have gone through some of the example of the screen. And first, we have obtained an aliquot from the originator lab. However, the expansion protocol is slightly different from our standard electroporation into STBL4 strain of E. coli. Their default protocol is using DH10 beta. However, this is a Reg competent cell, and our usually STBL4 is considered Reg A negative, which means that they are less permissive to recombination of DNA when there are highly similar segment. And that is an issue usually when you use lentivirus-based vector, which have like long tandem repeats. So we prefer STBL4. And they also inoculate their culture into liquid culture overnight. We usually spread our bacteria onto solid agar plate surface before scraping. But overall, we don't really see any skewing of the guide RNA distribution compared to the original aliquot. So both methods are valid, whether you inoculate the bacteria into liquid culture or you inoculate into solid culture first. However, we did see using spot check, just Sanger sequencing of the maybe 48 colonies from each of the two expanded culture, that we did see some recombination in the DH10 beta expanded library. And so we decided to move forward using our standard STBL4 expanded library instead. And so in the script of mapping the read, you have an option of whether to include one mismatch. So if you do that, you will have a higher mapping percentage. However, for consistency, we decided not to use the mismatch setting. We just use only the reads that are perfectly matched to both guide A and guide B. And you can see there is a little bit of a recombination between the two guides. So these are the guides where they have very defined guide pair. They should target the same gene, guide A and guide B. But when they decouple, each can be targeting different genes. So that would become very noisy. So we discard those reads. So in this instance, it's still a very tiny proportion, and we didn't really change the distribution of those guide pairs through expansion. So after lentivirus packaging, we also have some like pre-screen experiment that we need to do, and that is to express the dCas9 crab. And this is expressed on the mCherry vector, so that the lab then sorted by fax twice to enrich those cells. We did see recombination between the two guys, so we considered compensating that loss, potential loss of a recombination event by building in extra cells from the beginning. One thing that we need to be mindful using this library is that because each gene in the ultra compact version only have one data point, meaning that you only have one pair of guide targeting that gene, we do need a replicate in the experiment to be able to generate a p-value. And on the library vector selection, this is a puromycin and GFP dual selection marker, which is actually really nice. We can see enrichment of GFP positive cells along the puromycin selection period. So this is what this is kind of like a nice exercise to have for anyone who is new to running pool screens, where we can infect the library into cells. And this is our target cells throughout the screen during the titration. So we titrate our virus batch each time. And so as you start your puromycin selection, along the way, you can see an enrichment of GFP positive cells increases over time. And for the no virus control, after 5 to 6 days, you should have like almost no cells surviving at all. on the plastic. And so with two operators, we seeded the cells, accounting for our MOI of 0.3, and independently infected the cells, and gone through the cells will then go through puromycin selection. And we'll always make sure that we take an image of the cells and to make sure that we didn't lose any GFP signal. After 7 days, then we consider cells completely incorporated the library and see the cells to T1 and T2 treatment arms. So T1 is always the vehicle treatment, and T2 is the drug treatment. And for this particular screen, we are not using a too harsh of a condition. The drug that we give is more cytostatic. It's more at IC60. And you can see that at the endpoint that a lot of the cells are still GFP positive. So that is good, meaning that the guide RNA cassette is not being silenced, which can happen in some cases. But the cells are completely flattened down compared to the vehicle control cells. So this is a good kind of like exercise to have to make sure that we keep track of the cell phenotype over the treatment period. In terms of the amplicon enrichment, this requires a double-sided Sprite B selection. So the Sprite B select amplicon size is based on how much Sprite B you add to your amplicon prep. And so the first 0.5x selection will remove most high molecular weight, such as like genomic DNA fragment that's in there. And then the second 0.65x selection will retain the amplicon that are bigger than 300 base pair onto the beads. So you wouldn't be able to see the amplicon very clearly until you have done the double-sided bead selection. And another thing to consider is the input of gDNA per PCR. So in a lot of protocols, it's not exactly clear, but they will say, let's say, up to 10 microgram gDNA. And later on, we can see how like maybe the lower end of gDNA input per PCR is more beneficial for this particular library. And some of the exercise that is very good to have, especially for undergraduate students, this is kind of nice to see, like it's important to titrate your gDNA input per reaction. More doesn't mean better, because like more gDNA input, you can see that as we increase the amount of input per reaction, we start to lose the amplicon. So this is using a different library. This is using the LentiGuide library screen, where we have more reagent to play with. And that up to a point, your amplicon increases as your gDNA increases. However, at some point, what you have so much gDNA, maybe that there are some impurities from the elution that is inhibiting polymerase, or that there's so much gDNA is mopping up the primers due to non-specific binding. So it's a good exercise to always do a little titration to make sure that you will get an amplicon across all samples. Another thing that is not always explicit is the number of PCR cycles. In John Dench's Brunello library, they usually suggest 28 cycles, but we are able to titrate down the number of PCR cycles down to 24, 26. And it is really a balance, how strong your amplicon is versus like the background of like primer dimers and your non-specific gDNA in there. And this is an example done by some student, Alice Cantor. And the more PCR cycle you have, you may introduce skewing of the gDNA distribution, because guide RNA that may be GC rich can be preferentially amplified. And so after the screen, we have 3 sets of duplicates, and T1 are the freshly library incorporated cells that's been through puromycin selection. T1 is the vehicle treatment, T2 is the drug treatment. And after mapping, and this is using custom mapping tool on GitHub, we can see that the perfectly matched guide A and guide B, that percentage dropped a lot. So this is in line with what they observed as well, like 30% recombination. And just another point that having the index does not prevent recombination. The recombination will happen due to even like some of the shorter similarity regions between these elements. So it will still happen. And this is also in line with what the Broad had observed as well, which we'll talk about a bit later. But even with that level of recombination, we still have enough reads that are perfectly matched between those treatments for analysis. So we can see that actually like the replicates group together really well, and we can look at the T1 versus T0 treatment. So this is in the gene that, or the guide that are depleted in the vehicle treatment, and that gives us a sense of like how successful is our screen. And indeed, when you put the top hits into string database, it does nominate genes that are in the ATP respiratory electron transport or mitochondrial metabolism genes and tRNA biosynthesis. So these are very common essential genes. So that gives us the sense that actually there are signals in our screen. And because our collaboration have not been published, I'm not disclosing any gene targets, but here's the volcano plot between the drug treatment versus vehicle treatment that are compared to also the T0 sample. So anything in blue are the hits that are coming out of the proliferation control arms. So those are not our true hits that will follow, but the one that are in orange are the novel negative hits, and then the green are the novel positive hits that are more specific between the T2 versus T1, so the drug versus no drug comparison. And so what we can see is that, yes, so the one that are in orange on the negative side, those are the persistent drivers, meaning that if you knock out these genes or you knock down these genes, the cells won't be so persistent in the treatment. And then in green, those are potentially prognostic targets. So some of these are indeed tumor suppressors. So meaning that if you are, if the cancers are lacking this particular tumor suppressor, it may not respond so well to the treatment, that there may be persistence emerging faster than without mutations in those tumor suppressors. And so in terms of nominating the genes, our analysis was done on HR, since there's only like one data point per replicate. We, instead of using MAGIC, the mapping tool that is common for pool screens. Then we also looked at DECMAP to see whether they're essential or not, and prioritizing proteins that with known function or like secretion or cell surface, and also whether there will be ligandable pockets for drug design. So at the end, there are 25 anti-persistent hits that are nominated for follow-up. And indeed that there had been a dozen of, almost a dozen of the target was able to be validated individually in a follow-up array screen. So some of the off-target that we may want to consider are, so this is not a hit, but this is a tau MAPT, so a gene that is involved in Alzheimer's. You can see that like the guide target region, which is the promoter region that's annotated, there's also this antisense track. So sometimes we do see that when a gene failed to validate, there may be actually off-target effect that is targeting the antisense non-coding RNA. So that's something to keep in mind. And then let's talk a little bit more on the guide decoupling event. So a lot of it is from lentivirus packaging step that is an issue that is recognized in the Perturb-seq platform as well, because they have a very long common region between guide and barcode element. But guide decoupling can also happen at the PCR amplification step as well. And so John Dench has extensively addressed this. So that can occur as you increase the number of cycles during the PCR step. And so if we look at something that is similar for like the dual guide system, in the genomic DNA, if you use genomic DNA, like at 28 cycles, you would definitely see a lot more decoupling effect. So the coupling fraction are the perfectly matched guides. Those that are not matched are those 15% here. And so, and also, if you increase the amount of your input per reaction. And this is even more evident when we're using plasmid as input. The more input you have, you can see the perfectly matched guides start to decrease as well. So using this library for the dual guide CRISPR-I library, we try to keep our input per reaction to around 1 microgram per reaction. And so for the lentivirus step, where there is recombination happening, the template switching issue has been addressed extensively by Jonathan Weissman's lab, Britt Adamson, and also the Blaney lab. And there's a very detailed mechanism of like when that may be happening. The guide were swapped during a template switching event. And so their solution is to perhaps add a decoy vector during the packaging. However, the amount of decoy to the actual target vector for packaging is 9 times more for decoy. So you may actually lose virus titer by doing that as well. Or you can do arrayed lentivirus packaging, so there's no like multiple elements for guide to swap. And of course, that the longer your homology arm between the two elements, the more likely you will have decoupling happening as well. So if you are considering like Perturb-seq, those are known issues. So other way that you may want to go about this would be like using direct guide capture approach for crop-seq. And so now you can get the index version of CRISPR-A, CRISPR-I ultra compact library from Agene as 3 combined, or they also can be obtained individually. So how do we compare to like other libraries, for example, the single guide CRISPR-I library, Docetto from the Broad? If you have to consider just using the 1 element version, and you have to build in a duplicate and also account for 30% of guide decoupling event, the realistic library size is actually not that different. Of course, that the benefit is actually having 2 guides on the same vector and having a stronger repression effect. However, if you are using the 3-element compact library, then you do have a benefit still, even after considering the decoupling of the guide event. It will still be significantly smaller than the other version of CRISPR-I available from the Broad. And very quickly, I want to touch on the CRISPR-A screen. So this is something that we did for Robert Saxton, who is interested in engineering cytokines to have different functions, make them more specific for macrophages instead of T cells, in this case, IL-10. They can engineer ligands that are cell type specific. So they are interested in finding receptors for what we are called orphan cytokines, where the receptor for these cytokines are not completely understood and well characterized. Initially, they were looking for CRISPR knockout. However, in their purified ligand of interest, they did not find any cell line that are responsive to the cytokine. So we then settled on a CRISPR-A approach to activate the genes, and hopefully some of the receptors that are expressed can bind to their purified orphan cytokines of interest. And in the first instance, we're using PD-L1 and PD-1 as a proof of concept experiment to make sure that this actually works. So these are the common version of CRISPR-A. They usually bring in artificial gene activators that are borrowed from viruses or known transcription activators to the upstream of the transcription start site. The one that we settled on is the right library, just because the other libraries are divided into sub-libraries from genome-wide library, and that depending on how they bind the gene targets, for example, EGFR will be binned into the kinome library instead of the membrane protein library. So for this particular library, it's an activation library on the SAM system, which is this one here. And it does seem to target putative membrane protein proteomes. With this, they use a positive selection of max selection. So the magnetic beads already have bound purified PD-L1 proteins. And they can then separate the cells that are bound to the magnet, and they let them proliferate and do this again another time. So a total of 2 times selection using max beads, and that gives the cells potentially that are bound to these to this purified PD-L1 ligand. And in our screen that we did pull out PDCD1, which is the binding partner of PD-L1. This is PD-1 gene. That is the top hit. So at least in this case, we know that the system is working. And we are then able to help them use the same strategy to enrich cells that are bound to 3 different orphan cytokines. And so this is a really neat way of using CRISPR-A library. So one downside of CRISPR-A library is that like a lot of the existing CRISPR-A library to membrane proteins are not so functionally enriched for active guides. So the library size is much bigger. So perhaps like the 4 guide per gene CRISPR-A library, if they have like those guide design may be a bit more highly nominated compared to the existing one. So I hope that in this talk that I have stressed enough the importance of QC during experiment, and those are like very important steps. Like these are very long projects. So like if we don't do the QCs at each step, like it's pretty hard to figure out which step had gone wrong if there isn't any validable gene targets at the end of it. So it's important to spend the time to validate your experimental condition first. If you can have a positive control for doing that, that would be great. But even for the molecular steps, like say PCR, amplicon enrichment, it's important to do those QCs at each step along the way. With that, I would like to thank the IGI leadership, Jennifer Doudna, Brad, who's the director for the Institute, and Phil Ernoff, who's our Director of Technology and Translation, and thank my team, and Tess William, who did a lot of the CRISPR-I work, who's now a grad student at UC Davis. With that, I would take some questions.
Thanks very much, Alice. There's a real tour de force of all of the different CRISPR screening applications and technologies, CRISPR-I, CRISPR-A, and cancer. So anyone online for some questions for Alice?
CRISPR screen in T cells, yes, using sequential lentiviral transduction, Cas9 plus memory. I think that about 80% transduction. Yeah, 20% setup. Yeah, for T cells, it's all like primary T cells hard. I have run like we just completed this year a screen using primary human T cells. And of course that like because you are selecting, there's only like so long you can culture this cell, so it is really hard to condense that into, say, the 2-week period where you can actually culture the T cells and that they have the biology that's relevant. We found the slice method work quite well. So that is infecting the library and then electroporating the Cas9 enzyme. But you couldn't really do that with CRISPR-A or CRISPR-I. What we found is that you do have to titrate the library across multiple donors. Like 3 is not enough. We titrate them extensively between 5 to 8 donors. So those are the things that can make the screen expensive. And then also selecting donors that are more infectable compared to others. And then you only can, like practically, so initially we considered using fresh cells, like freshly isolated cells, but in the end it wasn't so practical. We have to use like frozen cells. So we would get a batch of T cells, isolate those CD3 T cells, freeze them down, and then we have aliquots of across multiple donors that we will optimize the condition for, both in terms of infectability and the actual biology for like staining optimization.
Can I ask, just along that line, how do you adjust for the activation that's necessary for T cell proliferation, or adjusting for those factors that are necessary to maintain primary cells like T cells that in themselves can also have a transcriptional effect? So, for me, I work with Tregs a lot and I have to activate them fairly regularly.
Yeah, so in our experiment, we had to activate them twice. So you have to activate them in order to put the library in. Otherwise, they're not so infectable. And then after the selection, before we add the drug, we'll have to activate them again because we're looking at say PD-1 biology. So the PD-1 surface expression diminishes over time, and then we have to like activate them again. But so the first activation we use beads, but the second activation we use immunocode. So different, like slightly different version of activation. I'm not sure if that answers your question, but those are the.
Is that 20% on second hit okay? Or should we improve on it?
Actually, 20% is the max we can get in our hand. So yeah, like we initially were only getting 10 to.
Can you put some selection like blastocytin for the second round?
Yeah, so we did we used puromycin. So
we did puromycin for Cas9.
Yeah, we use puromycin for library, but yeah, like it's the timing of selection is a bit shorter using primary cells. We at the end select them for 3 days who kind of like over the weekend. That fit in quite well with our workflow. And we built in one cell split within that 3-4 day selection period. Because if you don't split them, yeah, they just don't proliferate as well. But then we also find that towards the end in the screen, first of all, we did it across, like we did a screen in 2 different donors. And then we found that during the drug treatment period, if you split them, they actually have more cell death. So they're a lot sensitive to like even just how many times that you pipette them during those selection days. So they are definitely a lot more sensitive to death. And they're actually happier if you don't touch them during those final selection days for the drug treatment, but not the lentivirus selection. How do we take into account challenge of lentiviral library production, maintaining guide representation? So yeah, like the library production, well, there's several steps that you need to do, right? So like first of all, like you always make sure you have dilution plates from what you electroporated and grow out so that you can count by estimate like what's the total number of colonies you have on your master plate, and does those colony like cover say at least like 1000x of your library size, and then you will always QC at the end by NGS, and that will give you a distribution of your guide. And there are certain criteria that are very common in library construction, is that like did you capture all of the guides? Are all of the guides present? And then the spread of the guide distribution, like your top, I think it's like the top 10 and like the 90th percentile over the 10th percentile number need to be like below 5 or something. And so another one is when you have the from the dilution plate, always pick colonies to send for Sanger sequencing, because that at that point you will know like how much of those vectors are your religated vectors. Like there's always a plate where we do like no amplicon insert to see like are there any background from the cut vector. So these are like the like a lot more detailed QC step that you need to consider to make sure that if you see there's a high proportion of like religated vector or just mutation in the guide, or there's a like in one instance we saw just last month, there's like a 30% enrichment of one particular sequence, and that is very problematic. And so we have to go back and troubleshoot what that issue may be. So the issue in that sense is like there is a low diversity library, and something clearly need to be optimized.
Yeah, doctor, hello. Following on my question, the same. As I'm working on the Adgene Brunello library, which has 76,000 guide RNAs. So I did the amplification, the initial amplification of the library. But the next step is, the lentiviral production, packaging into the lentiviral vectors. So that step is important, whether we are able to package all of the 76,000 guide RNAs. So I was referring to that, lentiviral library production.
Yeah, so for that, you would always, well, there are guidelines from the protocol that are published,
Yeah.
like how much you do, and I always like to do maybe not double, like double maybe like a little bit excessive, but just like do more, because you never know. Like some people will build in, say, before they do the functional titration of the library, they also have like buy a kit from like Takana that have basically Roche have these kits that you can look at by qPCR how much lenti library you have in there. It's not a direct functional measurement, but like even before you do your functional titration, it's good to have that number so that you have in mind that, oh, like this batch is bad because like I only got like a tenth of what I had last time. That has a really good titer. Then you will go back and see, is it something gone wrong with the hex cells?